Hi, my name is Delatte. I'm a group leader in the Advanced Research Lab at Active Motif. And today I'm going to talk to you about a new technique that we developed at Active Motif and that was recently published called ABBS, or anchor-based bisulfite sequencing. So a few words on DNA methylation. So we know that DNA methylation is very important for genome biology and gene regulation. And in mammalian cells, cytosine can be methylated on its fifth carbon by enzymes called DNA methyltransferases, or DNMT. So there are two classes of DNMT. There is a unmethylated cytosines, and then maintenance DNMT as DNMT1 that will replicate methylation when cells are dividing. And so it's without saying that DNA methylation is very important for gene regulation, but also genome biology, such as DNA damage repair and DNA recombination. So it's not surprising, therefore, that DNA methylation is also deregulated in diseases. And a good example is a cancer, where we do see a global hypomethylation and a focal hypermethylation of tumor suppressor genes. It's also seen, for instance, in different diseases, neuronal diseases, such as Alzheimer's disease or Parkinson's disease, where we can find deregulated methylated biomarkers. So today we're going to talk about the techniques. So I want to give a little bit of an introduction of the different existing methods to map and to sequence DNA methylation. And so I distinguish two type of methods. So enrichment methods that give you a peak calling based approach, and bisulfite sequencing. So let's start with the 5MC enrichment. So there are different methods, but the two most used techniques are first MediPseq, or methylated DNA IP sequencing, and MiraSeq. So for MediPseq, DNA is extracted, genomic DNA is extracted, and then sonicated and denatured. And that's important because the methyl group is embedded in hydrogen bonds in the double helix. And so if you want to capture methylation, you will have to denature DNA. Then DNA is incubated with an antibody that recognizes methylcytosine, and it's bound on beads, washed, eluted, and DNA can be sequenced, and we can look at regions that contain DNA methylation. For MiraSeq, here we're using, instead of using an antibody, we're using a protein complex made of MBD2B and MBD3L1. And so this protein complex recognizes naturally DNA methylation and binds to methylated DNA, methylated CpG. And so these, if you will serve as baits that will be there, then captured on beads. And methylated DNA will be cleaned up and eluted and then sequenced. So these are a great method because they're relatively easy to do. But a few caveats is that they're non-base resolution methods. So usually the resolution is about the size of the fragmented DNA. So if you have a library at 300 base pair, for instance, you're going to have a resolution of 300 base pairs. Another caveat is that these methods are semi-quantitative, at least if we don't use a spike-in to normalize. And so we can say, for instance, we have more or less methylation in this or that sample. We have very few methylation or high amount of methylation, but we cannot say we have 5 or 10% more methylation without at least using a spike-in. So it's a inherently semi-quantitative method. Another type of technique to map DNA methylation is called bisulfite sequencing. And so in this case, DNA, instead of being incubated with proteins or antibodies, DNA will be incubated with a chemical, which is sodium bisulfite. And the most commonly used technique is called whole genome bisulfite sequencing, or WGBS. And so in that case, DNA is extracted, genomic DNA is extracted, and then reacted with sodium bisulfite. And cytosine that is unmethylated will be converted into uridine by bisulfite. And methylcytosine that we can see here with a little dots on top of it will remain as a methylcytosine after incubation with a bisulfite. So methylcytosine in a way is protected against deamination by bisulfite. And so after ligating a Illumina adapter, amplifying and sequencing, we can count CT SNPs that correspond to cytosines that are unmethylated, and Cs that are read as C will correspond to cytosines that are methylated. And so it's a method that can give you, in a way, a base resolution, genome-wide, very quantitative measurement of DNA methylation. Now, one caveat of this method is that it is extremely expensive. We need at least 25, 30x coverage for coding DNA methylation. And so that requires at least 1 billion reads per mammalian genome. So it's a very expensive method. So in order to find the best of both worlds, so between an enrichment approach and bisulfite approach, we developed what we call anchor-based bisulfite sequencing, or ABBS. So the protocol is as follows. We start with double-stranded DNA. And in this example, we have, for instance, a methylated locus on the left-hand side and an unmethylated locus on the right-hand side, all right? And we simplify that by saying there's only one cytosine, just for the example. So after bisulfite conversion and the slight sonication, these fragments that are now single-stranded will contain either a methylcytosine that was protected against deamination by bisulfite, or a uridine that was a cytosine that was converted by bisulfite. And so the most important part of the protocol is using specialized oligos that we call anchor oligos, where we use 5 random nucleotides and a terminal PPG, which is an analog of guanine. And so in the left-hand side, where we have a methylated cytosine, there is a perfect match between the PPG and the methylated cytosine. On the right-hand side, where there is a uridine, there is a mismatch between uridine and PPG. So the oligo is not fully hybridized and cannot really be elongated. So the selection of the methylation follows by using a clenohexamminus polymerase that will elongate the methylated fragment, but that will not elongate the partially mismatched unmethylated fragment. And so we can attach P5 and P7 adapters onto double-stranded DNA, and then pair and sequence and analyze the results using Bismarck or another package. So really, the goal of these methods is to redirect sequencing power to methylated regions. So a few results. We mapped 5MC with ABBS in E. coli, and we studied with the E. coli simply because it's a small genome, it's very cheap to sequence. And secondly, because we know exactly where methylation occurs. And in E. coli, methylation occurs at the second cytosine of the CCWGG motif. And so it is methylated by DCM enzyme. So a very important point is that when we grow E. coli and we start the culture, these CCWGG motifs are unmethylated. Now, when we reach a plateau phase, the second cytosine becomes methylated. So that's at that stage that we can harvest cells, perform the DNA extraction, and run the ABBS experiment. So now the results. As a reminder, again, CCWGG can be methylated by DCM into methylated CCWGG in E. coli. And so the very first piece of data I want to show you is this genome browser plot. And so we plotted the coverage of whole genome bisulfite sequencing as a reference, the coverage of ABBS, and then the methylation based on WGBS. We did that also in a DCM negative strain. And so if we start with the first track, we can see that roughly the whole genome bisulfite sequencing coverage is relatively flat. There is no real peaks. We can see some dips and valleys, but it's relatively flat, and there is no enrichment whatsoever. Now, with the ABBS coverage, we can directly see that there is a big peak in the middle of the track, And this peak corresponds exactly where methylation has been called by Bismarck here using methylation data. And that's exactly where we can find the CCWGG motif as well. Now, when we delete DCM enzymes in E. coli, we see that this peak disappears. So it's a good control to show the specificity of the assay. Now, this is a genome browser track, but we wanted to show you also some genome-wide data. So in order to do that, we plotted the big weak signal, so the read depth, if you will, normalized in RPKM around the CCWGG motif. And so we can see that we have a big enrichment of the bisulfite converted ABBS sample around the CCWGG motif, which makes sense because that's exactly where methylation occurs. Now, when we look at the bisulfite converted whole genome bisulfite sequencing, we don't see this enrichment. When we knock out DCM in E. coli, we can see that this signal disappears. So it's a good control to show that it's really specific for DNA methylation. And finally, when using a control motif, we don't see any enrichment for neither of the sample. Next, we looked at the enrichment of methylation and the coverage. So what you can see here is a scatter plot with the whole genome bisulfite sequencing intensity in RPKM on the y-axis and ABBS intensity on the x-axis, and two clouds of points. And so these points are either methylated clusters of cytosines, and that's the ones that are in blue, or unmethylated or non-methylated fragments. And so what you can directly see is that there is a bias. So ABBS seems to better enrich for these methylated fragments because we see that there is a skew of towards a horizontal line. And that's exactly what we want to see. So we're expecting to have an enrichment, a bias, if you will, towards methylated cytosines. So in conclusion, ABBS detects the expected CCWGG motifs, and ABBS provides better coverage for methylated fragments in E. coli. Now, let's start to talk about ABBS in human cells. So we did the mapping of 5MC with anchor-based bisulfite sequencing in K562 cells. And the very first piece of results is the FAST-QC that is spit out by the pipeline, if you will. And so the FAST-QC shows you the aggregation or the aggregate of nucleotides across the read. And so you can see that at the 6th position, there is PPG, or there is a guanine that is found here that is at 70%. And that makes sense because we are supposed to enrich for this guanine through the PPG on the oligo. And that's fixed. Now, what's very interesting is that the 5th position of this read is not fixed and is read here as a cytosine. And that totally makes sense because cytosine methylation occurs in CpG dinucleotides. So it's normal to see an enrichment of CpG dinucleotides at the beginning of the reads for the ABBS sample. And we can see that we have none of this enrichment for the whole genome bisulfite sequencing control. All right. Next, we looked at the ability of ABBS reads to cluster in methylated regions. So what you can see here, again, is a genome browser plot with whole genome bisulfite sequencing coverage, methylation, ABBS coverage, ABBS methylation, and also a MEDIPSI coverage, which again is a technique that enriches for DNA methylation using an antibody. So the first thing we can see is that the whole genome bisulfite sequencing coverage is flat. Again, there is no enrichment whatsoever, and we see a relatively flat coverage. And but when we look at DNA methylation that has been called by Bismarck bioinformatic package, we can see that there are regions that are highly methylated, and then regions that are very lowly or non-methylated, and they have the same coverage for whole genome bisulfite sequencing. Now, when we look at the ABBS coverage, what's very interesting is that the ABBS coverage parallels the methylation found in this sample. And so where we have methylation, we have high coverage. Where we don't have methylation, we don't have high coverage. And finally, we can also see that MEDIP-seq parallels very well the ABBS coverage. And so it makes sense because these are two techniques that enrich for DNA methylation. On the global scale, we can also see that ABBS intensity correlates very well with MEDIP-seq intensity, where we see a PCC of 0.93. And finally, we can look at metagenes, and the ABBS signal is clustered in the gene bodies, which is something that is known in the methylation field. Most of the bulk of the methylation is in gene bodies, and MEDIP-seq shows a similar profile, but we do not see any enrichment whatsoever for whole genome bisulfite sequencing. So in conclusion, we can say that ABBS provides increased coverage in methylated regions, and that is similar to MEDIP-seq. Now, we know that ABBS captures DNA methylation, but how accurate is the call for a DNA methylation? And so for that, we compared cytosine methylation for ABBS on the y-axis and cytosine methylation for whole genome bisulfite sequencing on the x-axis. And there's two things that we can see. The first thing is that there's a very good correlation between the two methods with a Pearson correlation coefficient of 0.99. And the second point I want to make is that we do see a bimodal distribution. So we see that there's lots of dots, if you will, at very highly methylated fraction or percentage, and there's lots of dots at very lowly methylated percentage. And so we know that, in methylation field, that methylation is bimodal. So very highly unmethylated and very lowly methylated, either or, and with a spread in between, but most of the points are either very highly or very lowly methylated. So in conclusion, there's an excellent agreement between methylation called by ABBS and whole genome bisulfite sequencing, and we do see this bimodal distribution. Another very good method to map DNA methylation is called reduced representation bisulfite sequencing, or ABBS. And in this method, genomic DNA is fragmented or digested with restriction enzymes that will recognize CG sites. And these CG regions will be cleaved, enriched, will create a library or ligate adapters, and then bisulfite converted. So after amplification and sequencing, we can obtain data or gather data, methylation data, in CpG-rich regions, such as CpG islands. But one of the caveats of ABBS is that it does not provide methylation data outside of CpG islands. So we wanted to see how well ABBS compared to ABBS. And so we looked at the 5MC sites identified with ABBS that overlap with CGIs and non-CGIs. And what you can see is that there is a good fraction of methylated sites that overlap with CGIs, but also a very good deal of methylation that are found outside CpG islands. So that's a good thing because it's not restricted to CpG islands. We can see here also that there is a fraction of 5MC sites that overlap with ABBS covered regions, but also a whole lot of sites that are not covered with ABBS covered regions. And so we can say that ABBS measures 5MC inside, but also outside of CpG islands. So that's an advantage of the ABBS compared to the ABBS. Probably the most important slide of this talk is this one. So we know that ABBS enriches for DNA methylation, and we guessed that it required low sequencing power. But so the way we tested it is by subsampling the FASTQ file, the sequencing file. And so we started with a full experiment, nearly 1 billion reads for whole genome bisulfite sequencing and 1 billion reads for anchor-based bisulfite sequencing. And then we subsampled for 50 million reads, 200 million reads, 100 million reads, and 50 million reads. And we looked at the number of 5MC sites that are detected per million reads. So it's a relative ratio. And what you can see directly is that we capture many more 5MC sites in the ABBS experiment compared to the WGBS experiment. And the second point is that at least in K562 cells, we get, if you will, the most bang for our buck at 200 million reads. That's where we get the most 5MC sites per million reads. So 200 million reads is, it's 5 times cheaper than a whole billion reads when doing Illumina sequencing. So it's significantly cheaper, and on top of that, we capture much more DNA methylation in ABBS than whole genome bisulfite sequencing. On the right-hand side, it's an absolute, level. So we're looking at methylated cytosine that are recovered in millions, and that's depending on sequencing depth. And so you can also see that we capture many more ABBS methylated sites with the ABBS sample than with the whole genome bisulfite sequencing sample. And if we take, for instance, even 100 million reads, for 100 million reads, we capture more methylation with ABBS than a almost full billion reads with WGBS. So we can safely say that ABBS requires at least 10 times fewer reads to detect as many 5MC sites as WGBS. Some unpublished data as well. So we looked at the ability of ABBS to detect differences in methylation between healthy and cancer tissues. So we used 4 tissues: lung, liver, breast, and colon. And we looked at normal versus tumor and clustered the sites that are methylated. So what you can see here are clusters and heat maps. And directly, we can see that we spot differences. So we can see, for instance, that there is a cluster of regions here that are hypermethylated in normal tissue and that lose methylation in tumor tissue. Or the opposite, we have a cluster of sites on the normal tissue that seems to be hypermethylated and that will gain methylation during tumorigenesis. So we do see differences in methylation between different tissues when using ABBS technique. And finally, ABBS is also able to detect methylation in cell-free DNA. And so for that, we used human healthy cell-free DNA, and we performed ABBS or WGBS and compared the data. And we can see that we have a very good correlation in terms of percentage of methylation with a PCC of 0.99. And I won't go into too much detail on this last piece of data, but we also see that we have a great difference between ABBS and whole genome bisulfite sequencing in terms of capture of 5MC sites depending on the threshold of coverage. And for instance, for 25x coverage, which is what is recommended by ENCODE, we do see that we capture nearly 41 times more 5MC sites in ABBS than WGBS. So in conclusions, ABBS provides an accurate measurement of DNA methylation percentage. ABBS detects 5MC genome-wide, and it's not restricted to CPG islands, such as RBS. ABBS increases coverage of 5MC by redirecting sequencing power to methylated regions. ABBS decreases the cost of 5MC mapping relative to whole genome bisulfite sequencing by 10 times. ABBS can detect methylation in cell-free DNA and also can detect differences in methylation between healthy and cancer tissues. And finally, it's an easy protocol that can be performed in one day. So the take-home message is that ABBS is a cheap alternative to whole genome bisulfite sequencing with enrichment comparable to MedIPSeq. In a way, it's a bit like a MedIPSeq with a base resolution. So our paper was published in Communication Biology 3 weeks ago. And as a fun fact, we were lucky enough to make the cover of the website. And that's thanks to Rai and his great schematic for the anchor-based bisulfite sequencing story. And I want to end by pitching the idea that methylation is not the only modification that we can, detect with ABBS. We could detect, for instance, hydroxymethylation if we're interested in looking at this modification. And that's something we're certainly doing right now at Active Motif. So with that, I'd like to thank the wonderful team that worked with me, Nate, who is the lead author on the paper, but also Jason and Joe Fernandez, and Ted DeFrank for their support and their great inputs on this paper and this project. And with that, I would like to thank you for your attention. Thank you.